Hello guys, welcome back to my channel, Plant Pathology Pathsala. So in this video, we are going to discuss about rice blast disease. So we will discuss about the history of rice blast disease, where it was discovered, how it is named, then about the causal organism, its etiology, then about the management practices. So watch this video till the end. Okay, so let's start. So first about the history part. So the rice blast disease was first recorded in China in 1637 and there it was called as rice fever. Okay. Then in Japan it was believed to be occurred or reported in 1704 and in Japan it is called as Imochibio. Then in Italy, it was reported in 1828 and it was called there as bruchon. So three name of the rice blast disease, three new name are now that are rice fever in China, then Imochibio in Japan, then bruchon in Italy. Okay. Then in India, it was first discovered in Tanjore district of Tamil Nadu in 1918. Apart from this, other names are rice rotten neck disease and rich man's disease. This rich man disease, this is a very common question asked in various competitive exams. So remember this, the rice blast disease is also called as rich man's disease. So five name we got here: rice fever, Imochibio, bruchon, rice rotten neck, and rich man disease. Next about the symptoms of rice blast disease. So the rice blast disease affect all the aerial part of the plant, and based on the stage of the plant, stage of the rice plant, the symptoms are divided into three categories, that is leaf blast, node blast, and neck blast. So, leaf blast generally occur at seedling stages, then node blast at tillering stage, and neck blast occur at reproductive stage. So, in leaf blast, here you can see typical spindle shaped or eye shaped or diamond shaped lesions which are with grayish center and brownish margin. Here you can clearly see that the spots are of spindle shaped with the end are little bit tapered end, and the gray is in center with brownish margin. And in the case of node blast, the node region generally, the blackening of the nodes occur. So, this blackening will extend both upward and downward up to 1 to 3 mm, and later it weaken or the node become brittle and it will totally break down. Okay. Then in the neck blast region, generally in the neck blast, this occur at reproductive stage, and so during grain filling stage, if it is occurring very early, then it may lead to white ear or no any grain filling will occur. That is, that will lead to empty grains. But in later stage, if it is affecting, it will, but substantially it will reduce the grain quality as well as grain quantity or yield. Okay. So if in the ARS mains exam or in any exam, if it is asked that write the symptoms of rice blast disease, simply you can mention it in three sentences. First, you will write leaf blast. So that you will write spindle shaped lesion with gray center and brown margin will be seen. Then node blast, blackening of nodes. Later it will break. Then in the node blast, in neck blast stage, so this is occur in reproductive stage, and at the neck region of the panicle. So neck means just like us, our neck is holding our head. Just like that, this neck region of the panicle, it is holding the whole panicle. So if the neck region will rot, the panicle formation will ultimately hamper. Okay. So in the neck blast, blackening of the neck region will occur, and that leads to white ear or empty grains. Okay. So this is all about the symptoms. Next, about the disease cycle. So the disease cycle include the primary source of inoculum, then secondary source of inoculum, and the mode of spread. So the primary source of inoculum means where the infection start. For rice blast disease, the primary source of inoculum is the infected seed and infected crop residues or infected plant residues, the stubbles. Okay. So this infected seed either carry the spores or infected this crop residue that is either carrying the mycelium. Then the secondary inoculum is the wind-borne conidia. Okay. And mode of spread is generally through airborne conidia. So air is the main agent that is spreading the disease. Here you can see the spore photo. It is of 3-celled pyriform shaped. So two septa are there, which is dividing the spore or conidia into 3-cell structure. Okay. This is about the disease cycle. So if any long question, if it is given, they are asking about the disease cycle. If you don't have time, you can simply write the primary source of inoculum is infected seed and crop debris. Secondary source of inoculum, airborne conidia. Mode of spread through airborne conidia. This is about the disease cycle. Okay, you can make a circular diagram also. Easily you can make the diagram. Okay. Then about the fungal etiology. So as we all know that the fungus that is causing rice blast disease is Magnaporthe oryzae. So Magnaporthe oryzae, it is having 7 pathotypes that is known to infect various crop plants like wheat and ryegrass. So many grasses family actually used to infect. So based on its capacity to infect the particular crop, it is given particular pathotype name. Okay. So Magnaporthe oryzae, pathotype oryzae is actually infecting the rice plant. Okay. So I will prepare a separate video on the Magnaporthe oryzae pathotypes. Okay. So we will discuss this later. So now on rice blast. So for rice blast, the causal organism is Magnaporthe oryzae, pathotype oryzae. Okay. Then about the pathogen or the fungus, it is a ascomycetes fungus, means it is of higher group order, means definitely it is having a septate mycelium. Septate mycelium means the mycelium with septation or septa or cross wall is present. Then it is producing asexual spore that is conidia or pear-shaped or pyriform shape, which is generally made up of 2 septa, making the spore 3-celled. 3-celled pyriform conidia. Okay. So here interesting structure it will form to penetrate the host tissue. So here in the photo you can see here that this three-celled conidia is producing a mucilaginous structure. So acid adhesive mucilage is produced from the tip of the conidia, and through that mucilage it attached to the host surface or the leaf surface of the rice plant. Then after this stable attachment, it produces a pouch-like structure. That pouch-like pressure bomb, you can say, pouch-like or balloon-like structure is called as appressorium. So appressorium help in penetration and attachment. You have to remember this. This is most frequently asked question, that the Magnaporthe produce appressorium and it is helpful for the fungus for attachment and penetration. So it's like a pressure bomb. Here about 8 megapascal pressure is created because of presence of a melanized membrane, and it produce the infection pack and finally penetrate the host cell. So as it produce multiple infection cycle, it is called as polycyclic disease. Okay, we will make a separate video on monocyclic, polycyclic and polyetic disease. Okay. Next, the favorable condition for blast disease development as well as the epidemic development. So it required a moderate temperature of 20 to 28 degrees Celsius and high humidity more than 90%, and leaf wetness is must, as it is essential for spore germination or conidial germination. So for every epidemiology study, this temperature and humidity, these are two are most important key features. Okay, so you have to remember this moderate temperature and high humidity. Then other predisposing condition that favor the blast development is excess application of nitrogenous fertilizer. I have already told this is called as rich man's disease. Why? Because of this, the excess application of nitrogen causing heavy infestation of rice blast disease. Apart from this, the high darkness period or enhanced darkness period induce the spore quality as well as sporulation. So generally for the experimental condition also, this 14-hour of light and 10-hour of dark cycle was preferred. Next, last but not the least, the most important management practices for rice blast disease. So, the most important, you can see most economic as well as most sustainable and commonly used practice for the rice blast management is use of resistant variety. So, resistant variety can be chosen as per the location of the location concern. So as the our government AICRP projects are there, multi-location trial are going on. So as per the location, many varieties have already released, so you can choose them. Like examples are IR 64, IR 36, Jabani, Jaya, Vijaya. So many varieties are there. Then mostly polygenic resistance is preferred because the rice blast, that is the Magnaporthe oryzae, the genome of this pathogen is very much plastic. It can easily adapt to the environment. So, the polygenic resistance is preferred over the monogenic resistance because it is more durable and it can sustain the boom and bust cycle. If you want more detail about these things, we will discuss it later. Okay, I will prepare a separate video. Then next most important practice is cultural method. So it includes removal of alternate host or the collateral host specially. So here the collateral host are the other weeds or grass family weeds like Lursia, then Dinebra species. So many weeds are there, Echinocloa. So whatever rice weeds are there that are generally act as collateral host for the rice blast disease or rice blast pathogen. Then the most important is split application of nitrogen fertilizer. Then to the chemical method here, as we already discussed, that's the primary inoculum for the rice blast is infected seed and crop debris. So we have to focus on the management of these two, because if we restrict the primary inoculum or primary infection, ultimately we can slow down the secondary infection. So the seed treatment is mostly preferred with captain, theorem or carbendazim or tricyclazole, 2 gram per kg of seed. Then spraying at nursery stage can be done with carbendazim. Then at main field, in the main field, Edifenphos, the carbendazim, tricyclazole, generate the triazole fungicide are most commonly used. Nowadays, strobilurins are can be used. Apart from this, antibiotics, antifungal antibiotics are can be used for the rice blast management. Example are casugamycin, blasticidine. You can mention these things. Then ultimately the biological method here also you can use seed treatment of the rice with trichoderma or pseudomonas, 5 to 10 gram per kg of seed. So these are the management practices. So if you have any other queries regarding rice blast, please mention that in the comment. I will try my best to answer each and every question. Thank you so much.